we were interested in understanding how different cells in the body actually use their genetic information in a different manner. And in the course of our studies, we discovered that the text of the DNA, the actual language of the DNA, can be annotated. It can be marked by chemical modification called DNA methylation. And it's this annotation, this marking, that tells this DNA, tells the genetic information in each cell of the body what should be read, which information should be read, and which information should be ignored. And in this way, this new level of language, DNA methylation, serves to regulate the genetic information. Now this turns out to have a tremendous amount of significance. Firstly, it has significance for understanding normal development. Secondly, it turns out that many of the changes we see in cells that occur in cancer are due to changes in this annotation process. That the information in the cancer cell is mostly the same as a normal cell, but the switches that control regulation have been modified.
Three decades of intense studies done by Howard Sider and myself resulted in the discovery of DNA methylation as a major regulator of gene function. DNA methylation is an important factor in our new understanding of disease and how to cure disease. In addition, DNA methylation is very important to understand stem cell biology and how do ordinary cells, which we call somatic cells, be reprogrammed to become cells that can divide to any tissue, which is now very recently, it was very important in designing new methods for therapy.
My lab has worked on DNA methylation, which is a modification of DNA, and it creates a fifth base. Everyone's familiar with the 4 bases in DNA. There is a fifth. What we need to know about a phenomenon like that, it's an actor, if you like, in search of a play. What one needs to know is where it is and how it's read. And we found ways of mapping where it was, and in the process we found a pattern of methylated DNA and non-methylated DNA, and those 2 sorts of DNA, it turns out, are read by proteins that recognize either one or the other and create either a gene expression unfriendly environment around the methylated DNA or a gene expression friendly environment around the unmethylated DNA. Now it turns out one of the proteins that we found, which binds to methylated DNA and creates the frosty environment for gene expression, is mutated in a human autism spectrum disorder called Rett syndrome. And that's given us an additional reason to be interested in, other than the blue skies reason. We've created an animal model of that disorder, and critically we showed that if you put the gene back in mice that imitate the human condition, after they have become sick, they get better again, which was a result that no one really expected. It was assumed that once you get ill with a brain disease, it's irrevocable. And so that creates the possibility that one will be able to generate a therapy for this disease, and I'd like to think we could do that.
Well, we have started with a very basic fundamental question, looking at the responses, the immune defenses of insects. Insects represent 80% of all living species, and they were known to be extremely resistant to microbial infections. So over something like 30 years, we have deciphered the immune response of insects, and from the effector molecules, which are novel antibiotics, to the signaling cascades and to the receptors. And in our discussions with colleagues from the medical field and from the mammalian field, it turned out that in man, in humans, and in mammals, much of these systems, which is present in a fly, is also conserved and plays a role in various sorts of defenses, first-line defenses, but also second-line defenses, that is to say adaptive immunity, by stimulating the adaptive immune response. And this has relatively important implications, as my colleagues, particularly Professor Akira, my co-laureate, have shown, namely in improving the systems of vaccination, in blocking inflammation, in fighting autoimmunity, in fighting allergy, and so on.
Our group identified the microbial component recognized by individual toll-like receptor family members and established the role of toll-like receptors as pathogen sensors. We reviewed a whole picture of toll-like receptor signaling pathways. Now many compounds which stimulate toll-like receptors are promising for development of effective vaccines, cancer immunotherapy, and treatment of allergies.
I began my research career studying the epidemiology of diarrhea and the relationship between diarrhea and malnutrition. Diarrhea was then the leading cause of child death in the world, and diarrhea in children still kills 1 million children a year. This relationship was soon found to be bidirectional. That is, diarrhea leads to malnutrition, and malnourished children have more diarrhea and it's more severe. We hypothesized that zinc deficiency might have a role in this relationship. Severe zinc deficiency that was discovered 30 years prior to that was found to have frequent infections, stunted growth, and hypogonadism. And we thought this sounded similar to what we were seeing with diarrhea and malnutrition. So we began a series of randomized controlled trials in children in low-income countries. Ultimately, it was discovered that zinc-supplemented children had lower rates of diarrhea, also lower rates of pneumonia, less mortality, and also that diarrhea could be treated with zinc and reduce the severity. The findings in regard to the benefit of zinc in reducing childhood infections and mortality were used by the World Health Organization and UNICEF to make a recommendation in 2004 that all children with acute diarrhea be treated with zinc as well as fluids. And this recommendation is now being implemented in more than 75 countries around the world.
One of the most exciting features of this incredible revolution in molecular biology and human genetics has been the recognition of two very important facts. The first is that we share so much of our genetic material with all living creatures and actually plants and other organisms on Earth. But the other side of it, and that's what's similar, is that every single human being is unique. There has been never anybody like you before, and there never will be anybody like you again. That particular quality, that uniqueness of the individual, has really been a source of much of the work that we've done, to focus on that particular patient who has a unique clinical presentation, which when studied can allow you to elucidate the gene, and then open up pathways that open up very unique approaches to treatment. Let me give you a few examples. There are small numbers of patients around the world who feel no pain. These particular individuals are given us an opportunity to define a critical gene involved in regulation of pain that is leading to new approaches in treatment of pain in the general population. There are some other rare families in the world who have no good cholesterol and have cholesterol studded on their mucosa. This has led to approaches to understanding how HDL is generated and also how cholesterol is regulated in different cell types that surprisingly is also leading to understanding about cholesterol in the pancreas, cholesterol in the brain, and approaches to think about modulation of this for treatment of diabetes. And then there's Huntington disease, where patients dying of this particular currently incurable disorders, where elucidation of this pathway is shedding light not only on Huntington pathways, but on pathways very relevant for other disorders like Alzheimer's. So from these rare patients who have shared their stories, their blood, their DNA, their support with us across this country and across the world, we've been able to define pathways and genes that are really being able to be translated to new approach to treatment, not only for these patients, but for patients with many disorders in the general population.